Alright, so today we're going to look at what's called a karyotype, and we're going to also look at mutations that happen within the chromosome. We've already talked about gene mutations with the last unit. Now we're going to look at whole chromosomal mutations. Alright, so we ended here the other day with the fertilization, haploid cells becoming a diploid cell, and for humans, we have 46 chromosomes, remember, and we can do a map of those chromosomes, and that's what a karyotype is. Okay, so looking at a karyotype, this is an actual picture, a map of the 46 chromosomes in a human cell. Okay, we have two different types of chromosomes. So the first 22 sets. So here on this picture of a male, you have 23 sets of chromosomes. These first 22 are called autosomes because they're automatically there. They're the same for everybody, for all humans, those 22 sets of chromosomes. Set 23, down here at the bottom, are your sex chromosomes. For females, you have XX. For males, you have XY. It's not really a Y, but it looks different, and it kind of looks like a Y under a microscope. But when you look even deeper, you see it's just a tiny X. It's a smaller version. Okay, and guys, we don't have all that extra chromosome on there like girls do because they're genes that we don't need. Things for from motherhood, genes for that we don't have to have. So that's why our chromosome is a little smaller. That's that Y chromosome. All right. Now, looking at some things that can happen and things we can tell using these maps and different disorders, diseases, problems, we can have an incorrect number of chromosomes. Now, don't have 46. We have too many, too few. Causes problems. For instance, here we have Down syndrome. It's one that's commonly heard of, where we have an extra chromosome on the 21st set. All right. We call this trisomy. The prefix tri means 3. So trisomy 21. So it's at the 21st set of chromosomes. Now, this causes issues. Right? It can cause problems with the child because it's not right. There's too many chromosomes there, too many gene pairs. We don't have pairs anymore. We got 3 of them. Okay. Looking further, that trisomy means 3 copies, or we could have monosomy, which means 1. It's all because the chromosomes did not separate correctly when they went through meiosis. Bad things can happen. You're talking millions of cells. The odds of your body messing up 1 or 2, yeah, it's pretty good because you're talking millions of times this happens. Okay? And age, your health, all these things play a role in all this stuff and how it happens. Okay, when they don't separate correctly, we call it non-disjunction. All right, so we have non-disjunction. Sorry about that. Non-disjunction here. Non means not. All right, dis is opposite. Junction to join, so does not unjoin correctly. Okay. Means that you can either have too few or too many. You're going to have both. If you have non-disjunction in this one single cell here, remember it's going to make 4 copies that are all different. Well, if we have trisomy, we have 2 that have 3 of them. That means they had to come from somewhere. So that means we're going to have 2 that have monosomy, just 1 copy. All of these are messed up. They're all wrong. They don't work right. And we have problems with all of them. Okay. All right. Some other forms of issues with the wrong number of chromosomes. We have trisomy 18. Remember that tri means 3 at the 18th chromosome. That's called Edwards syndrome. That's what this is. You see we have a bunch of different things that go with this. We have misshapen ears, misshapen head, small mouth and nose. You have a long torso, different foot pattern. Normally your feet are flat, though theirs are flexed. It means that they're constantly on their tippy toes. And the one that's most noticeable is the fingers. When you make a fist, your fingers are supposed to all roll together. Well, for folks with Edwards syndrome, their pinky and their pointer finger overlap the middle two fingers. Not something they can control, not something they can physically make right. It just happens. Okay. Patau syndrome is trisomy at the 13th chromosome. Monosomy, when you only have one, usually those zygotes don't make it. They usually you have a miscarriage. They die before birth because you're lacking a partner for that chromosome. Those genes cannot be expressed. They can't be shown. So if that's, say that's the gene for your circulatory system, well, you're not gonna have a heart or vessels being made because the genes aren't there. And so you get that. Usually those types of issues with zygotes, they don't ever come to fruition. The baby's miscarried before that happens. Okay. All right. Some other things that can happen. Some things that do happen. Non-disjunction of sex chromosomes. That means that whether you're male or female. Okay. So it involves the X and Y. The 23rd pair. It can be deadly, however, many people have this and they live. Okay, so we have Klinefelter syndrome. This is the double X chromosome. Right. Double X, which either means the mom gave two X's, or the dad gave an X and a Y. Don't know whose fault it is. It just happens where those sex chromosomes don't separate during meiosis, and the new zygote gets two from one of the parents. All right, there's a lot of things that go into this. You look at it, they don't know if they're male or female because they have two X's, which makes them female. They have an XY, which makes them male. So their body doesn't know what they are, male or female. All right. 47, that number, because now they have an extra one. So they have 47 total chromosomes, and you'll see that in both of these two coming up as well. XYY males, all right, means they have an extra Y in here. They got an extra Y from somewhere. It happened. You won't notice it. It won't be noticeable. No physical effects. So you can't look at the person and know they have this. All right. This usually affects their learning capabilities because there's a lot of extra hormones being produced, things that cause the brain to not work correctly. All right. You have a triple X female. All right, because triple X. You could be this and you will never know. There's no effects. Some of you right now could be this, ladies, and you never even know it. It doesn't affect you. Now, can it affect your offspring? Yes, because when this separates, some of the eggs are going to have two X's. Some of the eggs are going to have just one X. The odds of you passing that double X down to your offspring is pretty high, which means they could end up being Klinefelters. All right, that's a possibility, or they could be another triple X child. All right. Monosomy X, Turner syndrome, means that you only have one X. That's it. You don't have a 23rd pair. You have one sex chromosome. This causes many, many issues because you don't have the other genes and the other X or Y to pair with this in making your sexual components of your body. All right, chromosomal mutations. These are similar to gene mutations, but they affect more than just one thing. Okay, so we have deletion. Deletion is where a whole chunk of the chromosome is lost. Okay, so you take out a piece of a chromosome, not just one gene. You're taking out a chunk, which means you cause genes, multiple genes, to be missing. Okay, usually this, it is fatal. People can be born with these mutations, but they don't live long. All right, so Karyotypes, that's an example here. Portions of chromosome number 5 are missing. It's a deadly disorder. Children usually don't live very long after they're born. When I say very long, it could be a year, it could be 2 years, but they're not going to get to adolescence or get to adulthood. Duplication. A fragment joins another chromosome. Okay, so for instance here, this is Fragile X syndrome. All right, this X chromosome has over 700 repeats. This right here is just 3. So this is a normal chromosome. We have A, B, C, D, E, F, G, but you notice you got D and E right here. Well, in duplication, that chromosome now can have one set, or here you see it's got 1, 2, 3, 4 sets. If that happens 700 times, that X chromosome is going to be significantly different than the other one. The other X is not going to have all this. It's not going to pair up. Those genes are going to not be expressed because there's no partner for them, and that affects everything after that point as well with expression of genes. Okay. Translocation. Translocation. Moving, if you know what the word locate means, when you have a location, that's a specific point, specific place. Trans means to move, so we're moving locations. All right. A portion of one chromosome is exchanged or swapped with another one. If you notice in this karyotype, it's been colored so you can tell the different chromosomes. The chromosomes have been dyed so you can see it. If you notice right here, there's a little tip of this number 9 that's white. It shouldn't be. It should all be red. Where's it at? Well, if you look at 22 down here, that one has a red tip on it. That piece of this one and this one have been swapped. Now, it's going to affect the genes because there's no partner for that on the other chromosome. There's no white area on this other chromosome in the number 9 pair. There's no red area in this one. So you're going to have defective genes. All right. Acute myelogenous leukemia is an example. All right. The exchange between these two non-homologous chromosomes causes their blood cells not to work properly. Well, blood cells are important. Red blood cells, white blood cells, carrying oxygen to the body, fighting off disease, all that is compromised now. It's different. It doesn't work right. So these folks get sick a lot quicker because their immune system, their fighting cells, their white blood cells don't work right. And that's one example of how this can happen. All right. Inversion, to invert, think about your math class. If you have, 1 over 2 and they tell you to invert it, that means to flip it over. 2 over 1. Well, that's what happens with your chromosome. A small portion of it breaks off and it flips over. So if you look here at the example, we have a normal chromosome that would have GHI. Well, if it breaks off and inverts, it flips over to IHG. Well, these chromosomes, if this was a pair, they don't match anymore. All right, so gene A, that matches B, matches C, matches D, E. Those all match. F matches, but then, whoa, wait a minute, G and I doesn't match. If it doesn't have a pair, it's not expressed. So these three genes then would be gone. They don't get expressed. Causes issues. Okay, the genetic material is going to get read backwards. Doesn't work right. Okay, that's inversion. Insertion, pretty simple. Inserting something. We are putting something in that is not supposed to be there, and it's from another chromosome. So we have chromosome 4, chromosome 20. Significantly different. One's much bigger, a lot more genes than the other one. But this chunk here gets broken during meiosis and gets moved. And now we have 20 and 4 are both affected. There's no gene to match up on 4 in that homologous pair. There's an extra gene that won't match up in chromosome 20. Causes problems. Those genes, if they don't have a partner on the other chromosome, cannot be expressed. That could simply, could be hair color, doesn't work. There's no gene for hair color anymore. You have white hair. It could be pigment. It's how we have folks who are albino. It means their gene for pigment doesn't get expressed. All right. This might not be deadly. It might be. It could be because of what genes, what parts of what chromosome get moved around. Okay. All right. So that's chromosome mutations and karyotypes. That's what we're looking at today. We will be back tomorrow with some new stuff, talking about Gregor Mendel and his work. And tomorrow's notes are going to be a little more in depth because it's tougher, tougher material. All right, that's all for today.